Should probably get started. So, good morning, everyone. Thank you for coming. My name is Attila Loshonci. I'm from the Department of Neuroscience here at the Medical Center. And this morning, we will spend a little time together during this lecture to review chemical synaptic neurotransmission and review some of its interesting and salient features. So I have to pass through this disclosure slide. There's no scientific content in it. Just discloses my relationship with the material I'm showing. But that's the first slide. So the chemical synaptic neurotransmission is really a fundamental way of how neurons communicate to each other in the nervous system. And really, what you see here is an image from my lab. And just to orient you, what you see in red is a cell in the brain. It's a neuron. The neuron is filled with a red fluorescent dye, so you can see the cell body and its various dendritic processes. And simultaneously, what you see in green are axons from another neuron that forms and approaches synaptic contacts on this red neuron. And the little punctas you see all over are called presynaptic boutons. And this is the site where the synaptic communication, chemical synaptic neurotransmission actually takes place in the brain. So you may appreciate a little bit more this picture if I tell you that this was taken in a brain, from an intact brain of a mouse. And the mouse was actually doing something, behaving and learning why this image was taken. So this tells you two things, that synapses are nice, and this is a nice image, and they're probably important. So if you look at the numbers, you can actually learn a lot from this image as well. It tells you that there are many more synapses in the brain than neurons. In fact, in the human brain, there are about 100 billion neurons and 1,000-fold more synapses, so 10 to 100 trillions of synapses. And really, an average neuron makes about 1,000 to even 10,000 of synapses on other neurons, and in turn receive similar number of synapses. So it's an astonishingly complex way of communication, and indeed this is the fundamental way of how neurons communicate in the brain. And what we're going to talk about and review today, what are the basic mechanisms of synaptic transmission. Synaptic transmission in general, as you will see, can be extremely reliable and can ensure very reliable communication between neurons. It is also, as we will see, it has directionality, so it ensures the information transfer from a presynaptic cell onto the postsynaptic partner in a way that this is not diminished, not attenuated, so it's a non-detrimental way of information transfer. But one of the most perhaps astonishing features of synaptic transmission that it's not static. If you would be able to look at synaptic transmission that takes place at these very boutons, you will see that the strength and reliability of synaptic transmission that can be changed as a function that of the neuron, which is called synaptic plasticity. And this is thought to be a fundamental mechanism of many processes in our brain, including cognitive processes such as learning and memory, and pathological processes as well, that how synaptic transmission is altered. So we'll talk about mechanism of synaptic transmission and some key forms of synaptic plasticity, how the strength of these synapses that can be altered in our brain in a controlled way. So let's first review briefly the sequence of events that takes place at the synapse that underlies this synaptic communication, this form of chemical synaptic transmission. So, as I mentioned, there is a presynaptic that element here, the presynaptic axon that forms the presynaptic terminal on a postsynaptic neuron. And during chemical transmission, an action potential travels down on this that presynaptic that element all the way to the terminal and triggers neurotransmitter release. As you can see here, neurotransmitters are concentrated in small synaptic vesicles, and these vesicles, they sit in the presynaptic terminal. And the presynaptic action potential traveling and invading the presynaptic terminal evokes release of the neurotransmitter through exocytosis of these synaptic vesicles. So neurotransmitters could be released. What is important that neurotransmitter release, this process depends on calcium, the intracellular calcium in the nerve terminal. And how it happens is that the presynaptic action potential depolarizes the presynaptic membrane and activates channels that sit in the membrane. These are voltage-gated channels, so they sense the change in the membrane potential, they open up, and they allow calcium to flow from the extracellular space into the nerve terminal, leading to a transient increase in the intracellular calcium concentration, and this transient increase through an intricate molecular machinery that can lead to that neurotransmitter release. So calcium is important in the in synaptic function at the presynaptic terminal. Later we will see that it is also important in the postsynaptic side to regulate synaptic plasticity. But let's go back to the next step of events. So once these neurotransmitter release, they pass and diffuse through this tiny junction between the pre and the postsynaptic membrane, and they can bind and activate receptors that sit in the postsynaptic membrane surface. And once they activate these receptors, they can lead, it can lead to change that in the excitability that of the postsynaptic neuron. In some forms of synaptic transmission, that leads to increased excitability of the postsynaptic cell, that leading to a depolarization of the postsynaptic membrane. And once this depolarization that reaches a certain threshold, it will then activate another type of voltage-gated channel in the postsynaptic membrane called voltage-gated sodium channels, and the activation of these channels that lead to a postsynaptic action potential. So what you can see here, that under some circumstances, this transmission is extremely reliable and non-attenuating, so the presynaptic action potential is now transmitted onto the postsynaptic cell. So these are the main sequence of events that during chemical synaptic neurotransmission at a synapse. So let's look at and let's take a closer look of these receptors sitting in a postsynaptic membrane that plays such a crucial role in synaptic neurotransmission. So the neurotransmitters activate two major classes of receptors. The one that we call the ionotropic TET receptors. These ionotropic receptors are ligand-gated TET ion channels. So what happens is that the binding of the neurotransmitter produces a conformational change in this macromolecule and opens up an ion channel pore. That pore is this ion channel that's part of the macromolecule, and this opening can allow ions, positively or negatively charged ions, to pass through this channel pore and can cause depolarization or hyperpolarization. In other words, a decrease or increase in that membrane potential, leading to a more depolarized or more hyperpolarized membrane potential, depending on the type of the transmitter and the receptor. So the other type of receptors that present in the postsynaptic membrane are called metabotropic receptors. These metabotropic receptors are peculiar in a way that they don't have an ion channel. They don't have an ion channel pore. They can bind the ligand with high affinity. And this binding, instead of opening an ion channel, can lead to an activation of intracellular second messenger, a second messenger cascade that can in turn indirectly regulate the activity of that ion channels in the vicinity or even far away from the cell. So the action of the ionotropic receptors is really fast, and importantly, because they generate depolarization or hyperpolarization, changing the membrane potential, this effect is generally localized to the synapse. In contrast, the metabotropic TET receptors, they act on a slower time scale. If you would compare, the action of the ionotropic receptors takes place less than a millisecond. It's extremely fast. It's one of the main components that ensures fast synaptic neurotransmission. On the other hand, the action of the metabotropic receptors is much slower. It can take place on a time scale tens of hundreds of milliseconds or even at minutes. The other important difference is that their action, because of this diffusible second messenger intracellularly, is not localized to the synapse. Their effect could be sensed farther away from the synapse itself. And their action is not direct depolarization or hyperpolarization, but as I mentioned before, they indirectly can act through ion channels, and this can lead to change in membrane excitability, change in the resting membrane potential, change in the threshold of the action potential generation, or it can even change, as we will see later, the shape of the action potential itself. So these are two major classes of neurotransmitter receptors that participate in that chemical neurotransmission. So let's go and briefly review some forms of synaptic plasticity, how we can change the strength of synaptic transmission. So one form of synaptic plasticity is called homosynaptic plasticity. It's homosynaptic in a way that the activity of the nerve terminal itself will alter its release strength and release probability. So in a way, the release probability is altered by the cell itself. And one very simple form of this homosynaptic plasticity is called post-tetanic potentiation. So what you see here in this schematic, in the upper panel, that is the pattern of presynaptic action potential. In other words, the stimulation pattern of the presynaptic element. You evoke action potentials at a low rate, and during a short burst here, that you deliver a high-frequency stimulation, which we can call tetanic stimulation. But let's look at what happens at the postsynaptic side now. If you measure the postsynaptic membrane potential, what you will find that during this baseline period, during low-frequency stimulation, that there is a quite steady amplitude that responds, and during this tetanic stimulation, the amplitude or magnitude of the membrane potential depolarization gradually that increases. This is what we call facilitation. But what is the most remarkable, that following the tetanic stimulation, this facilitation does not disappear. It lasts longer. We don't have a time scale here, but it can last several hundreds of milliseconds or at even minutes, leading to this post-tetanic potentiation in the postsynaptic response. And perhaps this form of synaptic plasticity can represent the simplest form of memory in some organisms, including perhaps vertebrates and mammals. So this is one form of synaptic plasticity, homosynaptic plasticity and post-tetanic potentiation. Another form of plasticity is called heterosynaptic plasticity. And this is called heterosynaptic because in this case, it's not the presynaptic neuron itself, but a third element that regulates the strength of the synaptic transmission. This third element is generally called the modulatory neuron, and this modulatory neuron can release modulatory neurotransmitters, as you see here, and this modulatory transmitter can act on the presynaptic terminal and can change the probability of neurotransmitter release. So one form of this heterosynaptic plasticity is called, as you see here, presynaptic inhibition. It means that the neurotransmitter, the neuromodulatory transmitter released here, does something at the presynaptic terminal that leads to the decrease of the synaptic strength. That's why it's called inhibition. And how it achieves this is, for example, through inhibition of these presynaptic voltage-gated calcium channels. And as I mentioned before, I told you that the calcium channels are critically important in the presynaptic terminal to regulate that neurotransmitter release. So the probability of neurotransmitter release depends on the magnitude and the time course of this calcium current in at the presynaptic terminal. So if you would inhibit these calcium channels, then the calcium influx that would decrease. And all in all, eventually, this would lead to a decreased postsynaptic response. What something is important to keep in mind, that we're not necessarily talking about a single event of neurotransmission. Instead, we average out many events that would take place, and on average, statistically, we would find that during presynaptic inhibition, the likelihood of the neurotransmitter release that would be lower and would lead to a smaller amplitude average postsynaptic response. So this is one form of heterosynaptic plasticity and a presynaptic inhibition. We can also get facilitation, so increased strength in the synaptic transmission, called presynaptic facilitation. In this case, a different type of modulatory transmitter is released by this modulatory neuron. And this can lead to an enhanced synaptic strength. And one way to do it is, again, has something to do with the presynaptic calcium influx. In this case, as you see here, during facilitation, the dashed line would be the normal waveform and magnitude of calcium influx to the terminal, and that's prolonged and enhanced. And we have more calcium in the presynaptic terminal, so we can have a more reliable release. What is interesting, actually, how this can happen, and the way it happened has something to do with the change in the presynaptic action potential. as I mentioned at the first slide, or the second slide, the action potential, the presynaptic action potential, which is in gray here, is the major source of depolarization that activates these presynaptic calcium channels. So if you would prolong this action potential wake form, you will have longer time to activate calcium channels, so you will have more calcium influx. And the way to achieve it, that is actually inhibiting channels that are responsible for the decrease, the descending phase of this action potential, in other words, repolarizing the membrane. These are called voltage-gated potassium channels. So if a neuromodulatory transmitter would be able to inhibit these potassium channels, that could lead to a prolonged depolarization, an increase in the duration of the action potential, which can in turn lead to a prolonged calcium signal in the presynaptic terminal, and consequently, all in all, ultimately, it will lead to an enhanced synaptic strength, higher probability of neurotransmitter release. So this is, again, another form, an example of synaptic plasticity, heterosynaptic plasticity, the presynaptic facilitation. So let's review briefly, and the one really well-known form of chemical fast synaptic neurotransmission, which is called the neurotransmission in the neuromuscular junction. And this is a really nice example because we know a whole lot about this synapse in the neuromuscular junction, and it also gives us an opportunity to review again and go through briefly the structural components again of a synapse. So that, as you heard before, there is a presynaptic terminal. And this terminal actually comes from the axon of a motor neuron that in the neuromuscular junction, synapsing that on the postsynaptic muscle at a specialization called that end plate. So this presynaptic terminal, you can see this that enlarged version in this schematic, has a few interesting components. One of them is mentioned here. This is the mitochondria, because synaptic neurotransmission that is a highly energy demanding process. It requires a lot of energy and ATP, so mitochondria are here to supply that. In addition, you can see here the synaptic vesicles, and synaptic vesicles are actually concentrated near these submembrane specialization, which are called active zone, and this is called active zone because this is where synaptic release is taking place. And you can also see that these presynaptic voltage-gated calcium channels are also localized and concentrated near the active zone, so they are in this crucial position to let calcium in, to interact with the release machinery components in the active zone, and providing a switch mechanism for synaptic vesicle exocytosis. So these are some more details on the presynaptic terminal. So on the postsynaptic surface, we will see you see these receptors in the neuromuscular junction. These are called acetylcholine receptors. So the neurotransmitter that is concentrated and released from the presynaptic terminal is acetylcholine. The particular type of acetylcholine receptor in the neuromuscular junction is called nicotinic receptor, because these receptors are also activated by a plant alkaloid, nicotine. So these nicotinic ionotropic acetylcholine receptors, they are enriched just right below the presynaptic specialization in the postsynaptic membrane. And the last component that we already talked about, these are further down in the junction fold, these packets the enrichment of voltage-gated sodium channels, and these are the channels that can sense the depolarization in the postsynaptic membrane, and once they reach action potential threshold, reach a certain threshold, they can generate the action potential that in the postsynaptic that cell, in this case, in the muscle. So, of course, this is the sort of kind of structural view, and indeed, we know a lot about the neuromuscular junction, and very clever experiments were done since the 1950s by Bernard Katz and Paul Fath, for example, directly recording, impaling the skeletal muscle fiber with a tiny, tiny glass microelectrode, so they can record the membrane. Sorry.
Sorry, I didn't mean to interrupt your train of thought. On the previous slide, though, why does it show on the presynaptic membrane calcium channels if they were releasing acetylcholine on the presynaptic membrane?
So the calcium channels are ubiquitous. They are in all presynaptic terminals, and they are the source of the calcium influx. So they let calcium in, and this calcium increase leads to the release of the neurotransmitter, the acetylcholine, from the presynaptic cells. So calcium channels are practically present in all presynaptic terminals we know of. The type of the neurotransmitter, acetylcholine and others, we will review later, it can vary from synapse to synapse. Is that...
That was the neuromuscular junction, but on the previous junctions that you showed with nerves that were joining together, I thought the presynaptic terminal was releasing calcium.
No, the presynaptic terminals releasing the neurotransmitter in a calcium-dependent way. So calcium increases internally in the presynaptic terminal, and that leads to the release of the neurotransmitter. So the calcium is not released by the terminal. The intracellular calcium level is very, very low in the terminal. It transiently increases during release, so it can trigger neurotransmitter at that release. So what happens during these recordings that Bernard Katz, they can stimulate the motor neuron axon, and they can record the postsynaptic membrane potential and see what happens. And this is a time scale. The units are in milliseconds, so it tells you that this process is very fast. So the dashed line here, this is called excitatory postsynaptic potential. And this excitatory postsynaptic potential occurs with a single motor axon at a stimulation. As you see, if you look at the y-axis, this is large in amplitude. The muscles sit at a hyperpolarized negative membrane potential of -90, and this EPSP, excitatory postsynaptic potential, in this case, it's also called end plate potential. This is about 70 millivolts in amplitude. Why is it important? Because it is reliably crossing the threshold. This is the horizontal dashed line, the threshold for postsynaptic action potential initiation. So once the depolarization crosses this threshold somewhere here, right, then the postsynaptic sodium channels become activated, and they can generate a large postsynaptic action potential, a full-blown action potential. So the transmission in this in the neuromuscular junction is highly reliable, that you find under normal condition that reliable transmission from the motor neuron to the postsynaptic muscle because of these large and robust excitatory postsynaptic potentials. Of course, this process could be changed and the process could be altered. For example, indigenous tribes in South America found out that there are certain toxins that they can use in their arrows to prey or to kill the prey or the enemies under other circumstances. And this is called curare. And the curare has an active ingredient, tubocuronin, and this active ingredient can bind the acetylcholine receptors and block them. So this is a blocker of the nicotinic acetylcholine receptors. So once curare binds to the acetylcholine receptors and blocks them, they cannot open anymore, so the amplitude of the excitatory postsynaptic potential decreases. And doesn't have to decrease all the way, this is important, it just has to decrease below the action potential threshold so that the action potential is no longer generated, the muscle won't be able to contract, which could be the cause of death in case of unforeseen circumstances. Yes, so that's the functional, that sort of physiology of the neuromuscular at the junction. So let's look at closely a little bit the structure of this nicotinic receptor. So the nicotinic receptor, the nicotinic acetylcholine receptor, that is a pentameric structure. What does it mean? That there are subunits, individual subunits that are that closely related. They called homologous subunits. What you see up here is one of these subunits. Each subunit in this case, as you see, has 4 transmembrane domain with intracellular loops and extracellular that N and C termini. And 4 types of this, the subunits that could be made, and 5 of these subunits eventually form this pentameric structure. In this case, in this nicotinic receptor, there's 2 alpha, 1 beta, gamma, and the delta that a subunit. What you can see here that these subunits in this pentameric structure, they surround the ion channel pore that's similar to staves of a barrel. So these subunits surround and form a pore, and this channel is permeable to positively charged ions, cations, that's sodium, potassium, and calcium. So that when the channel is actually opens, when it binds calcium, so binds acetylcholine, sorry. So the predominant ion that flows through the channel is the sodium from the outside to the inside, because it follows its chemical and electrical gradient. The concentration, sodium concentration extracellular is much higher, so it wants to go in the cell. Also, it's a positively charged ion, and as the intracellular the potential is more negative, it will flow into this direction, so it follows both the electrical and the chemical gradient. So this influx of sodium will lead to the depolarization, and this inward sodium current that will generate this excitatory postsynaptic potential and plate potential we talked about earlier. So there are some pathological conditions where these nicotinic receptors are altered. 2 examples here. One of is called myasthenia gravis, and in this case there are antibodies. Is an autoimmune disease during which antibodies are generated against the acetylcholine, the nicotinic acetylcholine receptors, leading to a decrease in the surface expression of these receptors and decrease in the strength of that neurotransmission at the neuromuscular junction. There are other sort of genetic causes, certain genetic muscle diseases. For example, this is called the slow channel syndrome or congenital myasthenic syndrome, which are caused actually mutation in the receptor channel complex itself. There are many more forms of this that you will hear later. So let's leave the neuromuscular junction for a while, and let's look at some of the similarities and then differences in neurotransmitter, in synaptic communication between the neuromuscular junction and now the synapses that in the central nervous system. And we can use the the stretch receptor reflects, as an example to illustrate the dissimilarities and the differences. So during this during this reflex, as many of you know, there is a stretch receptor sensory neuron that has its the cell body and its receptor in the muscle, in this case in the extensor that muscle quadriceps. And these that sensory neurons, these spindles, muscle spindles, could be activated by stretching this extensor that muscle by tapping the patella. And once this happens and this sensory neuron is activated, that can release again that synaptic neurotransmitter, excitatory synaptic neurotransmitter, onto a motoneuron, the motoneuron that sits in the ventral horn of the spinal cord. So excitatory neurotransmitter is released directly on the motor neuron, that again that innervates that extensor, that muscle. In parallel, however, we have to somehow prevent the antagonistic that muscle from contraction, the flexor muscle. And how it's done is that the sensory that neuron also makes synapses on an inhibitory that neuron, and this inhibitory neuron releases inhibitory neurotransmitter called GABA or glycine on a motor neuron that in turn innervates the antagonistic muscle, keeping and preventing it from contracting. So, however, when we look at the properties and contrast the properties of this of neurotransmission, excitatory or inhibitory in the spinal cord, in the central nervous system, we would find that really the amplitude of these individual excitatory postsynaptic potentials generated by an individual sensory neurons are relatively small. So a concerted and synchronized action that from many of these sensory neurons are required to activate the motor neuron. And this is a general feature, I would say, that in the central nervous system, individual synapses are fairly weak. We talked about there are many of them, trillions of them, but what would happen if they would all be strong? I think it could lead to uncontrolled excitation or uncontrolled inhibition. Instead, generally speaking, the strength of the individual synapses are much weaker in the central nervous system. So this leaves room for both plasticity and sort of that regulation of synaptic strength through the interaction of the individual inputs. And it's exactly the same for the inhibition that we talked about, so that the sensory neuron also innervates and synapses on interneurons, these interneurons that will excite the motor neuron for the antagonistic muscle. And in this case, now inhibitory postsynaptic potentials are generated through the action of inhibitory neurotransmitters, GABA or glycine. And again, this leads to the hyperpolarization, a decreased excitability of the motor neuron, in this case keeping it from contracting the flexor muscle. And in this case, you can see these are actually the predominant types, the major types of neurotransmitters that you find in the central nervous system: the glutamate, the major excitatory neurotransmitter, and GABA, and to lesser extent glycine, major inhibitory transmitters. That these later ones, GABA and glycine, decrease the excitability of the cell through hyperpolarizing the membrane. Okay, let's go back a little bit once more and look at the structure of these ionotropic receptor once again. We already discussed one of the major subtypes, these pentameric channels, the nicotinic acetylcholine receptors, 5 subunits. In this ion channel family, we can also find the GABA receptors and the glycine receptors, and also some subtype of serotonin receptors in the brain. So they are pentameric in structure. And the difference, however, between the excitatory nicotinic acetylcholine receptor and the inhibitory GABA and glycine receptor is such that these two, the GABA and glycine, they are permeable to negatively charged ion, chloride. And because the chloride concentration in the extracellular space is much higher, when these channels open, they let chloride into the cell. This is a negatively charged ion, so it's going to go into the cell and will hyperpolarize the cell. So this is what is behind the inhibitory action of GABA and glycine. One interesting feature, perhaps, of the GABA receptors that is that they have several modulatory sites. This is that where drugs that you learn about that act on, including barbiturates, benzodiazepines, alcohol, general anesthetics, and neurosteroids. Generally, these compounds lead to an increase in the inhibition that through the enhanced opening of the channel, so it leads to a longer duration and larger that inhibitory that current through the GABA receptors. And we know about, of course, the genetic disorders, mutations in these channels as well, including the glycine receptor. For example, one example here is familial startle syndrome, when there is a mutation in the glycine receptor that leads to abnormally that large starter response. So this is one major subclass of ionotropic receptors, pentameric channels. The other one is the ionotropic glutamate receptors. These are also ionotropic cation channels, so they let positively charged ions, sodium, potassium, and calcium through, but these channels, in contrast to the other family, these are tetramers, so there are 4 subunits as you see here, and then 4 subunits surround the ion, the channel pore, and their subunit structure is also different, as you can see in terms of the number and position of the transmembrane domains. So, as I mentioned, glutamate is the most important and widespread excitatory neurotransmitter that in the central nervous system, and it acts through 3 types of that ionotropic that glutamate receptors. One is, and let me review those briefly. And these receptors are named after ligands that they have high affinity for beyond the glutamate. These ligands they are not present that in the CNS, but they could be used under that experimental laboratory conditions. So one is called the AMPA type glutamate receptor, the other one is the kainate that receptor, and the third one is the NMDA type ionotropic glutamate that receptors. There are a few interesting and important feature of these receptors, so that the AMPA receptors are located predominantly in the postsynaptic membrane, and indeed they are the major source of the depolarization that during fast excitatory synaptic neurotransmission. So when we talk about fast glutamatergic transmission in the central nervous system, we primarily talk about AMPA receptor mediated transmission. And the kainate receptors are predominantly located in the presynaptic membrane, can regulate calcium influx and neurotransmitter that release. So, and these receptors, most of these receptor types of AMPA and kainate receptors, that they permeable to the sodium ions, but they are not much permeable to calcium. So there is this third type of receptors, the most interesting one, I would say. These are called the NMDA receptors, and NMDA type glutamate receptors has a few interesting peculiar features. And one is that, as you see over there, these receptors permeable to calcium as well. We talked about the role of the calcium in the presynaptic terminal. There is a major role of calcium in the postsynaptic site, and it is one of the major sources of the calcium into the postsynaptic cell at the synapse is through the NMDA receptors. The second interesting feature is that actually it requires a another ligand in addition to glutamate, a co-activator, glycine, to open. The glycine levels, however, and in the background calcium level that in the CNS is normally high enough to activate, to sort of bind the receptor, so the activation of the NMDA receptor still largely depends on glutamate release. And the third feature is probably the most interesting, that there is magnesium, extracellular magnesium, that can bind and block the ion channel pore under resting conditions. So extracellular magnesium can act as a plug that can sit here that in the pore and prevent the calcium and prevent ion flux. So there is this is called extracellular magnesium block, and what is that interesting that this block is sensitive to the membrane potential. So the depolarization, transient depolarization of the membrane potential that can push this magnesium out. So it can relieve this magnesium block. Depolarization can remove the block, so the channel can become functional, active, and that can allow that ion through. So what you want to remember here, that because of these features, the NMDA receptor actually senses two things. One is the presence of glutamate. It has to bind glutamate to open, but it also requires depolarization. So it can act as a coincidence detector, a coincidence between glutamate release and sufficient depolarization in the postsynaptic membrane that is important for synaptic plasticity, as you will see in the next few slides. So let's just briefly go through the contribution of the AMPA and NMDA receptor-mediated currents to the postsynaptic potential. And we can do this, and it's important to note that most of the synapses in the central nervous system, they contain both AMPA and NMDA receptors. So generally, the current and the postsynaptic potential generated by a compound joint action of the AMPA and NMDA receptors, but their relative contribution is very different. And the normal resting membrane potential, which is minus 70 millivolt in most of these cells, what you see here, there is a fast and rapid and large amplitude AMPA receptor-mediated excitatory postsynaptic potential, while the NMDA receptor-mediated component is almost negligible. And why is that? You already know the answer, that at this hyperpolarized membrane potential, there is a magnesium block for the NMDA receptor. So despite they bind the glutamate, they cannot open because at this hyperpolarized membrane potential, magnesium that prevents them from doing so. So there is a small NMDA component. What happens then if we inject some current into the cell and depolarize the membrane potential, let's say to minus 40. In this case, the two things happen. The AMPA receptor component becomes slightly smaller, and that's because the driving force for sodium to pass through the AMPA receptor current is smaller at more depolarized membrane potentials, but it's still there, it's still a large component. But what becomes really salient now and large magnitude is the NMDA receptor-mediated component. And that's exactly because now at the depolarized membrane potential, the magnesium block is relieved, so at that NMDA receptor that are bound to the glutamate now can also conduct and become active. To separate these two components, of course, we can use pharmacological agents. There are components that act on AMPA receptors. This is called CNQX, for example. NMDA receptor blocker, this is called APV, that we will see in the next slide briefly. So if these NMDA receptors are mostly silent at hyperpolarized position per membrane potential, what is their role then? And in the 70s, there were extremely clever groundbreaking experiments performed by Bliss and Lomo, putting forward a model that since become a textbook information of how NMDA receptors can play a role in changing synaptic strength, changing strength now on a longer time scale called long-term potentiation. And they did this experiment in a structure of the brain called hippocampus, which is incidentally also a structure that's critically important in learning and memory. And since then, there became the links being forged between this long-term potentiation of the synaptic strengths and actually that hippocampus dependent that learning and memory. So long-term potentiation, or you see the acronym LTP, is important that in these cognitive processes. But what did they did? What Bliss and Lomo did, and they cut slices from the hippocampus, not really a cognitive structure anymore, but this allowed them to record the excitatory postsynaptic potential that in response to stimulation of the presynaptic axons. During this baseline, after this baseline period, then they give, again, a high-frequency tetanic stimulation, and what they found that under control condition, there is a long-lasting enhancement in the synaptic strength. So in the slice, in the brain slice, in vitro condition, this can last for several hours. Probably in the intact the brain, it can last for days or even longer, even for lifetime. So there is a long-lasting potentiation, and there's, again, this LTP is now thought to be one of the important cellular substrates for that learning and memory. But what it is also important that Bliss and Lomo found later, that this process depends on NMDA receptors. If you block NMDA receptors with this blocker, APV, then you can eliminate, you can block the long-term potentiation. So NMDA receptors are important in this process. So in the last two slides, let's just review potential molecular cascade that allowed this to happen and how actually NMDA receptors can play a role in this. So this schematic that shows you this sort of cellular molecular cascade that can lead to LTP, one predominant form of it at least. So what you see up here is the basal form of neurotransmission. At basal level, as we talked about, there is primarily OMPA receptor mediated transmission. The small amount of release, OMPA receptor can open and depolarize the postsynaptic membrane a little bit, but NMDA receptors remain mostly blocked and quiet, although they can bind at the glutamate. So now what happens that during this tetanic stimulation, the strong depolarization during this tetanic stimulation can open a lot of those OMPA receptors. And this depolarization can now remove the magnesium block from the NMDA receptors, leaving calcium inside the cell. And this calcium now can activate several intracellular messenger cascades, including calcium calmodulin-dependent protein kinases, nitric oxide, various kinases. And this process, the activation, the NMDA receptor activation, intracellular calcium influx, and the activation of the messenger systems can lead to enhanced synaptic strength in at least two ways. The one that you see here is the generation of the retrograde messenger nitric oxide that somehow leads to an enhanced synaptic strength. But probably the most predominant form is the insertion of new OMPA receptors into the postsynaptic membrane. And this leads to a higher density, high concentration of OMPA receptors. And this is how LTP can be expressed at this early stage, increasing the OMPA receptor density in the postsynaptic membrane that can lead to enhanced synaptic strength and larger postsynaptic responses. So in the last slide, we can ask, okay, what happens if we have LTP all the time in the synapses? We will potentiate all of our synapses, so that can cause problems for learning. So indeed, there is an orthogonal process in the other direction that we can weaken the synapses. This is called long-term depression, or LTD. And this, in many forms of the LTD, also depends NMDA receptors, as demonstrated here. APV blocks this depression of the synaptic response that occurs after now a low-frequency stimulation. Like in the tetanus, we talked about 100 hertz. In in LTD, we talk about a 1 Hz stimulation of the presynaptic cell that can lead to a long-lasting decrease in the synaptic strength. And again, some form of this LTD that depends on the NMDA receptors, and in the expression mechanism that's thought to be through actually removal of now AMPA receptors, so the opposite process what we discussed at LTP. Here we can remove these AMPA receptors from the synapse, so we can weaken the synaptic strength. And of course, one potential model that is that LTD can help to erase and remove unnecessary information. For example, it can help to remove memories that are associated with pathological conditions. So with this one, I will conclude, and thank you for your attention.
A couple of minute break, so I'll need the.
Oh, yeah. You got the next one?
Oh, okay. Is there someone who knows how this works so I can connect back to the computer?
So we haven't actually, we're having the training for the AV reps soon. So at this point, none of us are really trained or responsible, but I can do my best.
Where's the AV person?
I don't even think we have interest in
Thank you. Okay, I'd like to finish up with the second lecture of the morning. One of the problems with this room is that while the lecturer is lecturing, there is no clock to give feedback, inhibitory feedback, as to how to pace oneself. So we may have to designate one of you as a timekeeper. You know, you start waving your hand when there are 3 minutes left or something. 2-minute warnings. So now for something completely different, but there is a rhyme and reason. You're getting to learn how the nervous system works at a basic function, but that functionality requires a blood supply that's constant with virtual no interruption. It's the brain, the CNS probably makes use of about 40 to 45% of your entire metabolic output for its normal functioning, and that requires an integrated supply of blood. So I'm going to talk to you today primarily about the vasculature, arteries that supply various components of the CNS, but I will introduce this with a brief discussion of the meninges, brief discussion of the production of cerebral spinal fluid and how it travels through the subarachnoid space, and then most of the talk will then focus on the arterial supply of the brain. So the CNS has 3 coverings. Those of you who were at the Gross tutorial yesterday, and those of you who will go today and subsequent weeks, the brain is covered by a relatively thick and extremely tough membrane called the dura. It is located, of course, on the surface of the brain. Within the dura, there are large sinuses that carry blood towards the venous vein system back to the heart. These are large spaces within the dura. When there's a bleed, you'll have subdural hematomas. These can then result in pressure on the underlying brain and producing specific deficits. The next layer is the arachnoid. The arachnoid is a critical component. It's located here. It is in this particular space that CSF flows, and in addition, it's where the blood vessels, the arteries, and some veins are also located. So it's this particular place where, as I will discuss in the next couple of slides, where the blood-brain barrier actually exists between the blood vessels and the actual brain. And then finally, the very thin membrane that literally lines the surface of the CNS is called the pia. So the blood-brain barrier, of course, is a wonderful contraption that gives the brain its uniqueness, but it also makes it very complicated to apply therapeutics into the brain because of the extreme filtering system that are produced by the blood-brain barrier. And the barrier is primarily produced by the cells that make up the blood vessels themselves. So the blood vessels in the brain are different from the blood vessels in the rest of the body, in that they have extremely tight junctions. These tight junctions, as I'll show you in the next slide, are comprised of a high density of cell adhesion molecules that basically form a paste that prevents water-soluble molecules and ions to enter into the CNS region. Of course, if you're a fat and if you're a gas, you have no problems getting through the membrane because of the properties of the membrane. So those are not blocked at all by the blood-brain barrier. So pharmaceutical companies that want to generate drugs that will gain access to the blood-brain barrier, one way is to somehow add lipid moieties to the molecule and have it enter that way. And if the intracellular environment of the brain can remove those lipid moieties of those molecules, you have your drug of interest gain access to places where you would like them to be. But these are very complicated and not readily soluble problems to generate molecules that will actually penetrate through the blood-brain barrier. So there are some other tricks that one can utilize. One can, of course, break down the blood-brain barrier transiently in order to get things in. But the most reliable way to get things in is to take advantage of the facilitated transport system that the endothelial cells of the blood-brain barrier have that will actually transport your molecule of interest through the barrier. And I'll discuss that in a moment. So this is what the blood-brain barrier looks like. Here's the blood vessel, the lumen of the blood vessel. Here are the walls of the blood vessel made up of the endothelial cells. These areas that are illustrated here are where the tight junctions are formed. So we're looking at a cross-section. So imagine this now in the z-axis going for long distances of very dense molecular structure that forms these tight junctions. The other components is a pericyte, which is a major source of the basement membrane that surrounds the blood vessel. And then finally, the end feet of astroglial cells within the CNS that terminate around the structure here. All of these components represent filtering devices that prevent the ready access of molecules within the blood to gain clear access to the environment of the CNS. One takes a higher view of what these tight junctions are. There are three major cell adhesion molecules. Their names are not important. These molecules are located on both membranes of the endothelial cells. So this is a homologous or homotypic cell adhesion interaction. The same molecules bind very tightly with itself on the other side, and thus forming this very tight, pasty-like matrix within the extracellular space that will prevent the free diffusion of molecules. These cell adhesion molecules are maintained in the appropriate positions by adapter proteins that link the cell adhesion molecules to the cytoskeletal elements. Thus, you have a tight binding of and placement of these structures on both sides of the extracellular space. So these then form the tight junctions that prevent this. So as I said before, if you're a gas molecule or if you're a lipid, you have direct access to get through these membranes, so this barrier doesn't really prevent you from doing it. But if you're an amino acid, or if you're glucose, or if you're water, for that matter, you have to get across. And the process by which that takes place is a series of transporters Some of these are facilitating transporters, and that is they just simply shuttle the particular molecule of interest down its concentration gradient, so that doesn't require any additional energy for that point. If you're an ion, you get to the other side by ion channels. So there are sodium channels, there are potassium channels, calcium channels. I'm just representing the sodium channels here. So glucose can get across. If you're an amino acid, there are actually there are three general systems, but there are two major ones. One that deals with the large neutral amino acids. That's through the L system. It's a facilitated transport that doesn't require energy. There is another system for getting the smaller amino acids and charged amino acids. Initially, it's facilitated, but in order to get it actually into the brain, you need to have energy, so that's done through a transport system that is sodium dependent as well as ATPase dependent, that shuttles the amino acid from the endothelial cell into the milieu of the CNS. These MCT transporters are important during development. There is a source for energy, carboxylic acid transporters. These also are activated during periods of stress or during periods of low caloric intake, like in famines. These are then the substitute transporters for providing sufficient energy for the brain to function under these stressful situations. Now, as an example of how one gets material through the brain, into the brain through the barrier, is an example of L-DOPA. As many of you know, it's used to help those with Parkinson's disease. Parkinson's disease is a disease of the basal ganglia where dopamine-producing cells degenerate. So to alleviate the symptoms associated with that, L-DOPA, a precursor to dopamine, is used as the precursor. It hooks onto the amino acid facilitated transport system transporter to get inside the endothelial cell. Unfortunately, the endothelial cells have, on the one hand, they can transduce L-DOPA into dopamine. So the enzyme for that change is present in the endothelial cell, so that's the good news. The bad news is that monoamine oxidase is highly expressed in these endothelial cells, and that will then break down the dopamine. So you're left, in the absence of any other addition, you're left with no additional dopamine in the areas that you want it. So when patients take L-DOPA, they also have to take an inhibitor of monoamine oxidase, and that then facilitates the transport of dopamine into the brain. And you'll hear more about this, of course, when we discuss basal ganglia. So I'll just leave it at that. So the blood-brain barrier, therefore, is a very selective system. And with the existence of transporters in the membrane of the endothelial cell, this filtering system allows for crucial molecules to enter into the CNS. Now, the CNS is also bathed by cerebral spinal fluid. So I'll just use this slide to briefly describe where it's made and how it's then transported into the extracellular component surrounding the brain. So the choroid plexus is a source of CSF. It's located in the lateral ventricle and the third ventricle. And there's a choroid plexus also located in the fourth ventricle. So this is where the CSF is secreted. The CSF then is then diffused through the ventricular system, which you saw briefly the other day, and we'll discuss a little bit in more detail in subsequent lectures and in a few slides that I'll show you here. The CSF then flows through the various components of the ventricular system. Recall that the cerebral aqueduct is a very narrow aperture for the CSF to flow through. So any space-occupying lesion in this region of the brain could close this, relatively narrow aqueduct, leading to hydrocephaly, which can cause damage to various areas of the brain. And in order to alleviate that, you have to put in a device to remove the excess cerebral spinal fluid in areas where it's accumulating, so it doesn't, to relieve the pressure on associated structures in the brain that are receiving the pressure from the extra fluid. The CSF then gets out into the subarachnoid space through apertures in the fourth ventricle. There are two medial ones called Magendie, and there's a lateral one called Luschka, so the M's and L's help you remember that. So through these three apertures, the CSF then enters into subarachnoid space, and in order to gain access to what your brain milieu is like, you want to take a bit of a sample of the cerebral spinal fluid. So you may want to do a tap and gain access to that. Fortunately, there are large cisterns in the subarachnoid space around the brainstem and in the lumbar cord. So there's the cistern magna that's between the medulla and the cerebellum. There's a superior cistern at the level of the midbrain on the dorsal surface. There's the interpeduncular cistern on the ventral surface, and then there's the pontine cistern on the ventral surface between the pons and medulla. Now, if you want to stick your needle in this area of the brainstem, I would do that, but you should have high insurance because this is a pretty delicate place to be doing that. You can be good, but sometimes you can make a little mistake, but you don't want to do that. So fortunately, the lumbar cistern is a large space for collecting CSF, and even if you do make a little mistake, it won't be that bad for the patient. But with practice, one gets. So I must reveal, as I mentioned before, I had a medical situation earlier in the summer, and it's July. It's when residents and fellows join teams, and of course they're being mentored by a senior physician, and if I was conscious while the procedure was done, and when the mentor yells, "You went too far," you know it's anyway. But so the lumbar cistern is large, but I recommend that you don't go too far when you try to tap CSF, at least in July when you're first starting to do these things. So the CSF then flows around the subarachnoid space. It then gains access in part through to the brain via the blood-brain barrier. I don't remember how much CSF is produced per hour, but it's a large amount, so the fluid has to be removed from the system, and that removal from the system takes place in the superior sagittal sinus up here on the superior portion of the brain through these arachnoid granulations. The pressure in the superior sagittal sinus is very low, whereas the pressure on the other side, where in the subarachnoid space where the CSF is quite high. So it's a pressure valve and doesn't require any energy, and so the CSF then flows unidirectionally into the superior sagittal sinus through these granulations located on the superior surface of the brain. And so if we go back a couple of slides, these granulations that I illustrated here is the one-way valve by which the CSF gains access to the general circulation. Okay, recall, of course, how the ventricular system is positioned. Here is the lateral view. Here is a view superiorly. It's C-shaped. The lateral ventricle is a C-shaped structure. There's a posterior horn that goes into the occipital lobe. The lateral ventricle is connected to the third ventricle through a foramen called foramen of Monro. There are a pair of these because there are two lateral ventricles, so those gain access to the unitary structure of the third ventricle. What forms the wall of the third ventricle? What structure forms the wall of the third ventricle? The thalamus, good. And then this hole that you see here represents the thalamic adhesion, which is the medial nucleus of the thalamus merging with the contralateral and the contralateral thalamus. The third ventricle then flows, CSF will then flow into the aqueduct of Sylvius. Where is the aqueduct of Sylvius? What CNS components does the aqueduct of Sylvius flow through? That would be the midbrain and then into the pons. And the fourth ventricle is located primarily in the pons and medulla. There is no real ventricular system in the spinal cord because it's a patent space called the central canal, but it doesn't act. There are no active processes going on there. Okay, so I'll now focus for the remainder of the lecture on the arterial supply of the CNS, and we'll start off with the spinal cord. So the spinal cord receives sources from multiple blood vessels. The more anterior, the cervical, for example, the cervical and part of the thoracic cord will receive its blood supply primarily from branches of the vertebral artery, which is itself a branch off of the secondary branch off of the aorta. The vertebral artery gives rise to the spinal, anterior spinal and posterior spinal arteries, which will then flow down through the spinal cord to supply the ventral part of the spinal cord or the dorsal part of the spinal cord, respectively. In addition, from the thoracic cord on down, there are branches off of the aorta which supply parts of the spinal cord. As illustrated in this part of the slide, you can see that some parts of the spinal cord have a relatively high density of blood vessel innervation, so to speak, and others are low. So this sort of suggests that certain parts of the spinal cord with infarcts are subject to certain kinds of diseases. So the soft points, if you will, are the cervical thoracic boundary, primarily where where infarcts can actually have some serious repercussions, like paralysis and loss of sensation. In terms of the supply itself of the spinal cord, there is a significant amount of in areas where there is sufficient blood vessel supply, there is a sufficient amount of collateral circulation that if one vessel happens to be occluded, it is compensated for by blood coming from a neighboring blood vessel. But once the components actually dive into the spinal cord itself, there isn't that much collateral circulation. So these territories that you see here would then be supplied by specific branches that are either on the anterior or posterior portions of the blood vessels. Okay, so let's now move to the rest of the central nervous system. And so the CNS from the medulla on up through the cerebral cortex is supplied by two general circulations, the anterior circulation that is mediated through the internal carotid artery, and then the posterior circulation that is mediated through the vertebral arteries. So if we focus now on the anterior system, we have the paired carotid arteries labeled number 1. These then come up from the aorta, and there are numerous branches that come off. There's the main middle cerebral artery that supplies the dorsolateral aspects of the brain that I'll discuss in a moment. There's the anterior cerebral artery that branches, say, off this aorta, and will subserve and supply the medial surface of the brain. There are these smaller branches off of the internal carotid, such as the anterior choroidal artery that we'll discuss a little bit later. Another important artery is the posterior communicating artery that connects both the anterior system with the posterior system, and that's structure labeled number 4. The posterior communicating artery exists bilaterally in most people, but there is a significant population that will only have one of these arteries. The posterior communicating artery takes place at the level of the midbrain, so branches off of the posterior communicating artery will supply that structure. The posterior communicating artery then connects the internal carotid with now components of the posterior circulation. And the major branch here is the posterior cerebral artery, which is another major artery that supplies the inferior surface of the brain, in particular the occipital lobe. That posterior cerebral artery is a branch, the rostral branch of the basilar artery, which is a unitary artery here along the medial surface, ventral surface of the pons. There are several major branches off of the basilar artery. At the most rostral point is the superior cerebellar artery, and as its name indicates, it will supply the superior aspects of the cerebellum. Recall that the cerebellum has like a wedge-shaped structure, so it's that superior surface that's supplied by the superior cerebellar artery. In addition, as I will be discussing a little bit later on, these arteries also supply aspects of the pons, and they're important blood vessels for reasons you will see in a bit. The basilar artery then gives off lots of branches that supply the pons, and as we get down closer to the medulla, there is another major branch that is sort of parallels the superior cerebellar artery, and that it's the anterior inferior cerebellar artery, AICA, A-I-C-A, and as its name indicates, will also supply parts of the cerebellum, but also will supply the more caudal aspects of the pons, which we'll discuss in a moment. Then at the pons medulla junction, we have the boundary between the basilar artery and the paired vertebral arteries, labeled 6 here. The anterior spinal artery come off pretty close to this junction, and then travel caudally to supply the parts of the anterior component of the spinal cord. A little bit more caudally is another major branch, the posterior inferior cerebellar artery, which will supply aspects of the cerebellum, as I just said, but also aspects of the medulla, which we'll cover in a couple of moments. And then in the schematic, you can actually, you can review this at your leisure. So a closer look at the posterior circulation. So we have the paired vertebral arteries. Each of them will be supplying branches of and supply aspects of the ipsilateral, of their ipsilateral CNS components. So the right vertebral will supply the right medulla, etc. You have the major branches, which then supply components of the cerebellum. As I mentioned, there is a superior surface of the cerebellum and an inferior surface of the cerebellum. So the eye in pica and in ICA will be subserving this component. Recall that there's also a ventral and dorsal surface, so those 3 arteries will be supplying both the dorsal and ventral surfaces of the cerebellum, as indicated. In addition, these arteries will also supply the dorsolateral aspects of the brainstem. So in the case of pica, it would be the dorsolateral aspects of the caudal medulla. In the case of ICA, it's the rostral component of the medulla and a caudal portion of the pons. And in the case of the superior cerebellar artery, it would be the rostral portion of the pons. But these will all be on the dorsolateral aspect. So the artery then branches off its source, either the basilar or vertebral, circumnavigates the parts of the brainstem, and only starts to branch on the lateral and then the dorsal surface of the respective structures. For the more ventral portions, you have these short circumferential arteries that come off the basilar artery or the vertebral artery itself to supply the ventral surface. Some of these have names, but you will learn about that when the time comes. Okay? Then as we get up to the midbrain, we have the superior cerebellar artery, as I mentioned before, and the other branch, which is the posterior cerebral artery, which will then travel and supply the areas of the cerebral cortex that I will discuss in a moment. This branching takes place at the pons midbrain junction, and as you can see, the third nerve, the oculomotor nerve, sort of exits in between the two arteries, the posterior cerebral and the superior cerebellar artery. So in the next couple of slides, we'll review some of the aspects of the circulation and where these blood vessels actually flow. So here, using angiography, the physician injects a dye and takes pictures in rapid succession so that the dye has not yet diffused very much from the areas of interest that one wants to image. And of course, one uses angiography to locate points where there may be a bleed, or sometimes it's also used, this form of angiography is used to identify positions of space-occupying lesions, since those would cause a displacement of the major blood vessels. So here we have the posterior circulation, the paired vertebral arteries. 6 would be probably the spinal artery coming off. It then joins the basilar artery. So if at the arrow, where in the central nervous system would this be located? What part of the CNS is at the arrow? Is it the midbrain? No. Is it the pons? It's the pons medulla junction. Okay, so this is where the paired vertebral join with the basilar. What structure then lies underneath vessel number 2, the basilar artery? It's the pons. So these branches that you see here would be then supplying the pontine structures. At this point, at the end of the basilar artery, you have the major branches coming off. You have the superior cerebellar artery coming off in this direction, and then the posterior cerebral artery then flowing posteriorly. So this image is basically through the chin towards the back of the head. So you can see the posterior cerebral artery continuing to the posterior portion of the brain, for example, the occipital lobe back here. Okay? Same is true on the other side. This is now an angiography of the middle of the anterior circulation. So the internal carotid has this circuitous course as it comes up to the point at the midbrain, where it then branches into its 2 major components. So this would be then a frontal view on this side. This would be a lateral view of the same dye injection. You can see the paired anterior cerebral arteries coursing through the medial surface of the brain here, whereas the middle cerebral artery first travels through the area of the insula that is deep. If you pry apart the temporal lobe from the frontal lobe in the lateral fissure, You have the insular cortex. So these loop-de-loops that you see here, which are part of the middle cerebral artery, is parts of the insular cortex as it travels along the surface of the subarachnoid space associated with that part of the brain. And then towards the end of the lateral fissure, it then emerges onto the dorsolateral surface, and you see some of those components. You can certainly see that here. It's a little bit more difficult to see here. Whereas in this lateral view, the anterior cerebral artery forms several branches. You can imagine what structure lies where 6 is. The artery sort of follows the course of the cingulate sulcus. So you have here the cingulate gyrus in number 7 and the corpus callosum in area number 6. And from your medial view of the brain, which you'll see in a moment, these are there following the sulci located in that particular component. And then you have the various branches that then emerge and come off it at various points, supplying either the frontal lobe, the parietal lobe, and as far back as the parts of the occipital lobe. This is magnetic resonance angiography. This is the technique of choice where you combine MRI with angiography, and this is primarily used for identifying the location of space-occupying lesions because you can take advantage of some of the structural features. So some of you can identify some of these features. Let's start with number 1. It's the internal carotid. And why do you think the internal carotid looks like a bar as opposed to just the circle that it actually is? Anyone want to take a guess? Some of you are making this kind of a finger. It's the way you would describe a circular staircase. But recall, recall this sigmoidal shape as it goes up. So these magnetic resonance imaging, it's a relatively thick slice. It's not a thin slice that you would normally use for fMRI, for example. So you're capturing this sigmoidal direction of the internal carotid. So that's what you see here. Then it branches into 2 major components: the anterior cerebral artery, going towards the medial surface of the brain, and the middle cerebral artery, going here. We have this blood vessel here located right at the midline. Tell you, you can see what it is. It's the basilar artery, so therefore it's lying above the pons. It gives rise to a major branch, the posterior cerebral artery, which is flowing back towards the posterior portion of the brain. So in the next couple of slides, let's examine, and this is sort of the critical aspects of the lecture for you as clinicians, because you will see in the emergency rooms folks suffering from a variety of strokes, and those strokes are produced by blockages of either branches of the major arteries or the major arteries themselves. So it's important to understand the territory supplied by each of the major cerebral arteries. So if we look first at the middle cerebral artery, as I mentioned before, it travels through the insular cortex and deep to the lateral fissure, and then emerges and then supplies the dorsolateral aspects of the cortex. So it supplies this area of the cortex. So it's parts of the temporal lobe, so the superior temporal gyrus and the middle temporal gyrus will be supplied. And then in terms of the other parts of the frontal lobe, both the frontal lobe, the precentral gyrus, the postcentral gyrus, parts of the parietal lobe. But you'll notice that the branches only make it approximately up to where in the homunculus of the motor cortex or sensory cortex would be the upper trunk. All right. So what are these blood vessels from? Well, those are blood vessels of the anterior cerebral artery that supplies most of the medial surface of the frontal lobe and the temporal lobe, and then comes up over the superior surface of the brain to supply the superior surface of the frontal and parietal lobes. Okay? The posterior cerebral artery, as I said before, supplies the inferior surface of the temporal lobe, so it would be the inferior temporal gyrus, and then underneath parts of the occipitotemporal gyrus. I leave out the parahippocampal gyrus because it gets a special treatment. It then continues on the inferior surface and then supplies most of the inferior and medial surface of the occipital lobe. The lateral parts of the occipital lobe, some of it is supplied by the medial the middle cerebral artery, as well as the posterior cerebral artery. So you'll notice back here in the back of the occipital lobe that there is a potential for collateral circulation. Right? You'll notice that at the superior surface of the brain, there is potential for collateral circulation where branches of these major arteries can actually coalesce and supply those parts of the brain. All right? So this represents where those collateral areas of collateral circulation, right? At the top here, it's between the middle and the anterior. Here, it's between the anterior and the posterior, and down here, it's the middle and the posterior. Okay, so these areas are sources of collateral circulation. So collateral circulation is good news, right? So if one of those branches happens to be clogged, the other can compensate. But it's also a bit of bad news. If those of you who are familiar with hydrostatic forces, if there's a sudden, it's basically Boyle's law, which P1 V1 equals P2 V2. If there is a sudden drop in pressure, the volume will have to be compensated for, and so sometimes you get infarcts of those blood vessels that are in these zones of collateral circulation and result in the collapse of those blood vessels, a response to pressure, and therefore lead to certain infarcts. So there's good news and bad news, and it's the balance of that evolution presumably worked on to present us with the circulation that we have. All right, so what structure lies above this artery over here? Well, since it gives rise to the paired vertebral, it's the basilar, and it would be the pons located in that region. So I've just discussed the superficial branches of these major cerebral arteries. In addition, there are penetrating branches that supply the deep structures. All of you are by now familiar with some of those deep structures, and the three most important are basal ganglia, thalamus, and internal capsule. All right, so those are critical structures that need to be supplied by these arteries. So there are a significant amount of variation between which arteries supply which parts of these internal structures, and that, of course, will have to be determined on a case-by-case basis. But as a general, the most general feature is what I'm going to describe now. So most patients will have this kind of arterial supply of the deep structures, but there will be variations in that. And of course, you'll be able to detect what the problem is using other potential methods. So here's just an example of what some of these deep penetrating branches look like. So here is a case of a deep penetrating branch coming off the anterior cerebral artery or the middle cerebral artery. They come off at basically right angles of those blood vessels. And in this case, as you can see, it will supply aspects of basal ganglia. So the most inferior surface of the basal ganglia will be supplied by a branch of the anterior cerebral artery, whereas most of the rest of the basal ganglia will be supplied by the middle cerebral artery. As you can see, since this is a section through the frontal lobe, you can see one of the limbs of the internal capsule, right, the anterior limb of the internal capsule. So it receives supply from the middle cerebral artery. Okay, so there are two dimensions that you have to consider. There's a medial to lateral dimension of these deep structures, and then there's an inferior and superior dimension to these structures. And different branches will supply different aspects. And of course, you're not responsible for this. So this, now you can stop writing or something like that, because this slide is complicated, but I don't really have a way of doing this other than to describe it this way with a summary slide located here, okay? So these are coronal sections through the brain. These are horizontal sections through the brain. And the basic take-home message is that the middle cerebral artery and the anterior cerebral artery will be supplying aspects of basal ganglia. The internal capsule will be supplied, yes? No, no, there's no anastomosis for the deep penetrating branches. The question was, is there anastomotic connections at this level, and the answer is there is none. So a blockage of one has consequences. So as I was saying, the basal ganglia are supplied primarily by the middle cerebral artery and the anterior cerebral artery. The anterior component of the internal capsule is supplied primarily by the middle cerebral artery, whereas the posterior limb of the internal capsule is supplied primarily by the anterior choroidal artery, which is a branch off of the internal carotid that occurs just before it splits into the middle and anterior cerebral arteries. All right, so there's the anterior choroidal artery, and it supplies the posterior limb of the internal capsule, and it also supplies the amygdala and components of the parahippocampal gyrus. So that's the structure labeled in red. Okay, the thalamus is supplied by the posterior cerebral artery. Okay, and that's going to be our canonical average patient, but recall there'll be difference. I don't know if you had a discussion of another component of the internal capsule, which is the genu. That is when the anterior limb and posterior limb bend. That bend itself, turns out that bend has the axons that are important for supplying the motor and sensory aspects of the face. So it's the descending cortical control of the face, both ascending sensory information and descending motor information. And in many patients, that area of the capsule is supplied by a branch of the anterior cerebral artery, but in other cases, it's the choroidal artery, the part of the posterior limb. So there's a lot of variation there. So we're as what we're defining canonical, that the anterior, this branch of the anterior cerebral artery is supplying the genu of the internal capsule. Since I started a bit late, I don't think I'll have a chance to finish these structures, but I want to quickly go over the two clinical cases that are at the end, just to see the importance of knowing which parts of the brain are supplied by which particular artery. So you can read along responsively, perhaps. The patient is a 50-year-old woman with a heavy cigarette smoker and had hypertension. and very suddenly developed right-sided weakness and numbness and inability to speak. So the suddenness of the onset suggests a stroke. It happens quickly, and the symptoms happen quite quickly. So there's right-sided weakness, okay? So that's motor. That's numbness. That's sensory. Notice it's right-sided. There's not any specification about upper, lower, middle. It seems to be general. And there's speech. Now, as the course progresses, you will come to appreciate that there are other parts of blood vessels that are supplying things in brainstem that can also produce a subset of these things. So if speech is left out, but the motor and sensory weakness, that tells you a piece of information that perhaps you should begin to look a bit subcortical. All right. So then this one, no speech and follows only simple commands and makes occasional incoherent moans. So there seems to be both a motor and sensory component that's involved with language. There are visual, the inability to make eye movements to the right, the face is drooping down, and the right arm and right leg are weak. Okay, so recall that I mentioned that arms and legs in the cortical area are separate. So generally you want to look for Occam's razor. Typically applies if those are philosophy majors. There must be some philosophy majors in this group. No philosophy majors? What is Columbia coming to? Occam was a monk in the 14th century who came up with a brilliant principle that if there are multiple explanations to explain something, the simplest one is more likely to be true. Okay, so you want to look for a single entity. Clearly, you know, a person could have multiple infarcts in multiple cerebral arteries. That's a possibility. But the usual thing is that one artery is occluded with a sudden onset. So how might you be able to explain sensory, motor, and speech deficits? Okay? So based on your thorough knowledge of brain function, where would these be located? Where would weakness in the arm, if it were in the cortex, where would weakness in the arms be? In the premotor cortex, right? You can't move, right? You're paralyzed. What about lack of sensation in your arm? Well, that would be the postcentral gyrus, okay? So it's on the dorsolateral surface. Where are the speech areas, right? There was Broca's area for speaking and Wernicke's area for understanding speech. Those are all located on the dorsolateral aspect, right? So how do you explain the leg weakness, right? Remember, I told you leg was on, you know, on the medial surface supplied by the anterior cerebral. So how would you explain it? How would you explain it, ladies and gentlemen? All right, I'm going to give you a hint, and you just work on it, all right? What about the deep branches? The deep penetrating branches of the middle cerebral artery. And now I'll leave that to you to figure out. So the indication would therefore suggest that this occlusion occurred pretty close to where the middle cerebral artery branched from its source of the internal carotid to give you this widespread change as opposed to a branch of the middle cerebral artery somewhere later down. Okay? And you can look at let me just do case number 2 because it sort of is the flip side. Okay, you can, sudden left-sided weakness, left facial droop, that's the face, weakness in the leg, left arm and leg, and no sensation in the leg. All right, so there's weakness in the left arm and the left leg. But notice what happens a little bit later on. The left arm recovers some function. So what we're left with is left leg, motor, and sensory. In addition, she has this strange alien hand syndrome, which is called. You know, her left occasionally would grab onto things without her being aware of it, etc. So this is an Oliver Sacks kind of story. And she couldn't localize her left arm in space. All right, so obviously we couldn't not add the actual symptom. It's an actual case. So you're at this level, you're at no point to be able to address where that is coming from, but you can appreciate a couple of things. First of all, it's specific. Her actual deficits are in the leg as opposed to any other body part. She does have a bit of a facial droop, right? So that might be accounted for by a branch of the anterior cerebral that supplies the genu of the internal capsule. So it's something that doesn't fit the rest of it, so you have to come up. So when you're thinking about these stroke-like occlusions, you have to take into account potential for both superficial branches as well as penetrating branches as contributing to the symptoms. And so this would be a case of an anterior cerebral artery being affected. And it turns out that the supplementary motor cortex, which lies in the frontal lobe just in front of the precentral gyrus, the primary motor cortex, is the source of this alien hand component. And you'll get to hear more about that as the course progresses. So the lesion, therefore, is here in the territory that's supplied by the anterior cerebral artery. Thank you for your time. I'm sorry I went a little bit over. A group of you will head up to the gross brain tutorial. Those of you who are going to those gross brain tutorials this week, you're a bit at a disadvantage since the scaffold upon which the material we're covering there isn't fully present. So those of you who want to come back again at later times, next week and a week after, can do so. Some of you pointed out a mistake in the first lecture. These are sprinkled here and there just for us to know whether you in fact are actually paying attention. No, it was just a typo, sorry.